Okay. So chapter 7 is the other side of the photosynthesis process, right? This is where we're taking the stuff that we make from photosynthesis and we're using it at a cellular level. This is how it is we harvest energy from biologically important molecules, such as sugars, proteins, and fats. When we talk about cellular respiration, however, really we're going to be talking about it in the context of sugars. Just keep in mind that you can do this with proteins and fats as well. We'll talk about those at the end of chapter 7. The reason that we use sugars as an example for cellular respiration is because it shows you all of the steps of cellular respiration. Some of the organic molecules that we're metabolizing, you don't go through all of the steps. But with sugars, you do. So cellular respiration is inherently an exergonic reaction, where we're taking something complex and we're oxidizing it into something less complex and releasing a whole bunch of energy. In fact, I want to show you that equation. So I'm going to start by showing you the photosynthesis equation so we can use it for the sake of comparison. Excuse me. So with photosynthesis, we said that we took 6 carbon dioxide molecules, 6 CO2s, and we added to it 6 H2Os. And we converted that into 6 oxygen molecules plus sugar, right? We said that this reaction right here was what? Oxidation or reduction? Oxidation. And this reaction here was reduction. Yep. Now, cellular respiration is the opposite reaction, where we're starting with a sugar, C6H12O6, and we're adding to that some O2, and we're taking 6 oxygen molecules to do that. So this is why, you guys, you start breathing a lot heavier when you're doing something aerobic, like running, or cycling, or swimming. Because the metabolism of sugar that you're burning for energy requires some oxygen. And from that, we're making 6 water molecules and, plus 6 carbon dioxide molecules, which is what we exhale, right? When we're done metabolizing, we breathe out, we're releasing a whole bunch of carbon dioxide. And we know that for us, carbon dioxide is a metabolic poison. Okay, so ultimately what we want to do is we want to take this and convert it into that. Is that an endergonic or an exergonic reaction? It's exergonic. So does that make it oxidative or reductive? Oxidative. So we're oxidizing sugar into carbon dioxide. And we're going to take these oxygen molecules and ultimately we're going to fix them into water, which we know would be a reduction reaction. So at this point, instructors often point out how sympathetic natural systems actually are, right? Because we have the stuff that's being created by plants being used by animals, and the stuff that's being created by animals being used by plants. Okay. Now, like photosynthesis, cellular respiration is going to be a series of redox reactions. You guys are going to see electron transport chains again, and you're going to see ATP synthase again. And y'all are going to see a cycle very much like the dark reactions again. And it's just a large biochemical pathway in which we're successively oxidizing and reducing molecules, picking up an electron so that we can pass it on to something else. So there are really two stages to photosynthesis. There are 4 processes that we'll talk about, with 2 discrete stages. The first of which is anaerobic, and the second of which is aerobic. All of us are capable of, for a short time, creating energy in the absence of oxygen. I don't know if any of y'all have run a bunch or cycled a bunch or been in a race, but there's that point right at the end of your run where you're going as hard as you can and you feel like you can't breathe enough to keep up with the demands of oxygen inside your body. That's where you move to an anaerobic stage. You're not actually able to breathe enough to metabolize sugar, but your body can still do something to produce a little bit of sugar, and that's anaerobic respiration, and we'll talk about that stage right first. Now this is the most primitive of all types of cellular respiration. Even the most primitive of organisms, those that evolved like 3.5 billion years ago, we think did this, anaerobic respiration. And there's still some organisms today that only go through anaerobic respiration. Things like yeasts and bacteria are anaerobic. So when y'all make bread, and that bread rises because of yeast, the reason that it's doing that is those yeasts are fermenting the sugars that you're putting into the breads, and they're anaerobically making some carbon dioxide and some sugar. And when alcohols are made, those yeasts that you use in the production of alcohol are anaerobic. They aren't requiring any oxygen, but they're using that sugar to make a little bit of energy for themselves and producing carbon dioxide. And then the second stage is an aerobic stage. This actually requires some oxygen. And so what we do in the aerobic stage is we take one of the products, which is something called pyruvate, and we completely break it down to make a whole bunch of carbon dioxide. And by breaking down pyruvate, we're able to generate a whole bunch of ATP. Questions so far? So this is just broad strokes overview of the process of cellular respiration.
Aerobic uses pyruvate to oxidize sugar.
Pyruvate is an oxidated state of sugar. We're going to take sugar and we're going to make pyruvate, and then we're going to take that pyruvate and completely break it down to make ATP and some electron carriers. Okay. Now all of this is occurring in and around the mitochondrion. Our anaerobic stuff all occurs outside the mitochondrion, and then as soon as we move to our anaerobic stages, the Krebs cycle and the electron transport chain and chemiosmosis, that all occurs inside the mitochondrion.
Can you repeat that?
You betcha. The anaerobic stage of cellular respiration occurs outside the mitochondrion, and all the aerobic stages occur inside the mitochondrion. Okay, so the redox reactions that we're using are the process by which we take our sugar and our pyruvate and completely break it down into those 2 critical elements: energy, ATP, and electron carriers. And we're actually going to have 2 electron carriers. We're going to have something called NAD plus that becomes NADH, and something called FAD plus that becomes FADH2. NADH and NADPH serve exactly the same function, as does FADH2. These are just shuttles for electrons. Okay, so the first stage of cellular respiration is something called glycolysis. So what I'm going to do is I'm going to blank out the PowerPoint, and I'm going to diagram it on the board. And you guys are going to find that glycolysis is a relatively simple process. Now, what I'm going to want you to know about each of these successive stages within cellular respiration is their purpose, which I'll write on the board, and their products, which I'll also write on the board. Okay, so we want to know how much of each thing we're making at each stage of cellular respiration. So give me just a minute to erase photosynthesis. When I diagram glycolysis, I'm going to be diagramming a glucose molecule. And y'all will remember that in the past, when I've drawn a glucose molecule, it's been a 6-member ring, right, with 5 carbons and an oxygen. It's been relatively complex. And sugar occurs in that form, but it also occurs in a linear form. And that's how I'm going to draw it for cellular respiration. I'm going to draw glucose as a series of 6 connected circles. And each of those circles represents a carbon atom. So our glucose molecule consists of 6 carbons, right? It's C6. We want to take that 6-carbon molecule and we want to start breaking bonds. That's important because, remember, bonds represent nothing more than potential energy. So by breaking bonds, we can harvest that energy and make ATP from it, okay? So the first stage in cellular respiration is actually a little bit of an investment. What we want to do is we want to take this incredibly stable molecule and we want to destabilize it. And the way that we destabilize it is by attaching some phosphates to it. So we take 2 ATPs and we drop off their terminal phosphates. So that's going to look something like this. So we've invested 2 ATPs so far. And what that's done is it's taken our stable 6-carbon molecule and it's destabilized it because we've stuck 2 large negative groups on either side. Now, within photosynthesis and within cellular respiration, there are 2 ways in which phosphorylation can occur. And phosphorylation is the process of taking a phosphate and sticking it on to a molecule. The first type is called substrate level phosphorylation. And substrate level phosphorylation is where you take a phosphate from one molecule and you put it onto the next molecule. So I'm going to write that on the board over here. Substrate level phosphorylation. And phosphorylation, this is going to be the common term between all of our types of phosphorylation events. Phosphorylation just means that you're putting a phosphate on something. That's important because that's energy. So substrate level is transferring of phosphate, the transfer of phosphate from one molecule to another. Do you guys see how that occurred right here? That this was a substrate level phosphorylation event? Because we took a phosphate from ATP and we stuck it onto glucose. So that was substrate level phosphorylation. Now, it becomes so destabilized that that 6-carbon molecule is actually going to break into 2 3-carbon molecules. So that's going to look something like this. We have a phosphate up here and a phosphate down here. That's the second step in glycolysis. But that's not all of the step. One thing that you guys need to keep in mind is every time a bond breaks, electrons are going to be released. Yep,
How did
Sophia.
it get reduced to 3? I just, yeah, like it was at 6.
How do we
and
go
I was-
from here to here?
Yeah.
So what we've done is we've stuck some negative charges on this one molecule, and those negative charges want to be apart from each other. And so the easiest way to think about it is this 6-carbon molecule splits apart and becomes 2 3-carbon
Oh.
molecules. Does that make sense?
Yeah.
Okay. Now, when we break a bond, we release some electrons. And here at this stage, we're actually releasing 2 electrons. And so what happens is NAD plus comes along and it picks up those electrons and becomes NADH. And that happens twice. The problem is we now have 2 carbons that used to be attached to something that no longer are. And they want to fill those bonds. So what happens is the other type of phosphorylation event, where we get an inorganic phosphate, or something that's not a phosphate that isn't attached to another molecule, it's just kind of floating around in the cytosol, and it attaches to either end of those 3-carbon molecules. That's the second type of phosphorylation event. This is oxidative phosphorylation.
So when you drew that phosphate, is that a C next to it?
It's an I, so lowercase I for inorganic. So guys, let's stop for just a second. Remember how you felt when we started the light reactions of photosynthesis? And it just was incredibly overwhelming because there was so much crap going on. You're probably feeling something similar right now, but I promise you, this is all going to make sense because we'll go through it multiple times. So just like, ride the wave right now, and eventually it'll all make sense, I promise. So don't be overwhelmed at this point. But let's talk about oxidative phosphorylation. Oxidative phosphorylation is where a phosphate is taken from the cytosol and attached to a molecule. What is cytosol?